Hello students, welcome to Mahesh Angadi Science YouTube channel. Well, we are discussing one chapter here that names as cell cycle and cell division. Okay, in the previous video we already discussed regarding interphase of the cell cycle. Yet now, if you have not seen that video, just visit to the I button and go through that video, because that is the basic for this video. So first watch that video, then come to this video. Okay, here in this video we are discussing regarding M phase, that is mitosis phase. Okay, here M phase refers to what? Mitosis phase. That means here we are studying mitosis right now, a kind of cell division called mitosis. Okay, then what is M phase? What is M phase? Let us understand what is M phase. See here, M phase, it is a phase of actual cell division. See here, M phase is it is a phase of actual cell division. Here the actually cell is going to get divided, and it is the most dramatic period of the cell cycle. This point is going to be very important. It's the most dramatic period of cell cycle. Okay, this is because so many changes or so many reorganizations will occur in the cell cycle. Okay, so many reorganizations between the cell constituents occur in this stage. That is why it is said as most dramatic period of the cell cycle. Okay, then here the daughter cell, see here, a daughter cell receives same number of chromosomes as that of the parent cell. That is why it is also known as equational division. Okay, equational division. Why it is named so? Because here daughter cells receiving same number of chromosomes as that of the parent cell. Daughter cells receiving same number of chromosomes as that of parent cell. Okay, that is why it is called as therefore it is called as equational division. Okay, this point might be asked for your examination. Why mitosis or M phase is also known as equational division? So here the point is, the daughter cells are receiving same number of chromosomes as that of the parent cell. Hence, it is also called as the equational division. Let's say, for example, if parent cell is diploid, then daughter cells, how many daughter cells are going to be there? 2 daughter cells are going to be there at the end of mitosis. Then how many chromosomes they will receive after this kind of cell division? Remember, if parent cell is diploid, even daughter cells are also going to be diploid. They are receiving the same number of chromosomes as that of the parent cell. Hence, it is said as equational division. Okay, if the parent cell is haploid, then the daughters will receive how many number of chromosomes? Okay, then see here again the funda is same. If parent cell is haploid, the same number of chromosomes are transmitted to the daughter cells. Hence, daughter cells will also receive a haploid set of chromosomes. Okay, so that's why it is termed as equational division. And in M phase, okay, M phase, there are 2 further phases. Okay, M phase is basically divided into 2 more phases. Okay, that is the first one, karyokinesis. Okay, means nuclear division. karyokinesis, which is also known as nuclear division, and one more division is there called cytokinesis. Okay, that means cytoplasmic division. Yes, this is how the M phase is further divided. Mitotic phase is further divided into karyokinesis, that is nuclear division, because in the mitosis first nuclear division takes place. Nucleus will get divided into two daughter nuclei. Okay, that is why it is said as karyokinesis. Karyo refers to nucleus. Remember this word karyo refers to nuclear, kinesis refers to division, so it's going to be a nuclear division. Whereas cyto refers to cell or cytoplasm, kinesis refers to division, so it is a kind of cytoplasmic division. At the end of cell division, even cytoplasm will also equally distributed to the both daughter cells. That is why always M phase end with cytokinesis. Okay, and in here in karyokinesis there are further four phases: prophase, metaphase, anaphase, and telophase. Here one important point we need to remember is, of course there are some key features for each and every phases, but no clear cut lines are drawn. Okay, no clear cut lines can be drawn between the phases. Okay, that point we must remember. Okay, now let us understand the karyokinesis. Okay, now let us understand what karyokinesis, and all those phases comes under karyokinesis. Okay, as I already said, karyokinesis is further divided into four phases. Karyokinesis is further divided into four phases. Among them, the first phase is prophase. Okay, first phase is what? Prophase. Second phase is metaphase. Then third phase is anaphase, and last phase is telophase. You can remember all these phases by this formula PMAT, like that. PMAT, one kind of mnemonics you can make. Okay, here P for what? Prophase. P for what? Prophase. M for metaphase. A for anaphase, and lastly T for telophase. Like this you can easily remember all the four phases of the karyokinesis. Now what we will do is, we will study the first phase of karyokinesis, that is prophase, with the help of suitable diagram. Okay, whole phenomena you can remember with the help of that diagram. Okay, let us understand what is prophase now. Always remember that I am giving you one trick. One trick here is, see here, prophase, it's the first phase of cell division. Okay, then when metaphase comes, you can easily write the first point, that is metaphase is the second phase of the cell division. Then anaphase is the third phase of the cell division. When it comes to telophase, what you are going to write? I think you might be telling that it's a fourth phase. No, you can't tell it like that. You can tell it as a final phase of the cell division, right? Yes, see, so prophase. Okay, prophase, this is the first phase of cell division. Okay, first phase of mitosis or M phase. Okay, first phase. Pro, here the word pro refers to first, and phase refers to stage or a step. Okay, so this is the first phase of the M phase. And when we look into the microscope, when we observe under the microscope regarding prophase, what kind of changes are observed there? Let us see with the help of a diagram. Okay, let us see with the help of a diagram. See here, let us assume this is a cell, and this one here. Can you identify this cell organelle? Okay, yes, you can identify this, because in the last video or in while telling regarding S phase. During S phase, this centrosome got duplicates. Okay, this is one centrosome. It got what? Duplicated in the S phase. Okay, this is what? Centrosome. Okay, it is made up of two centrioles. Each centrosome is made up of how many centrioles? Two centrioles. Okay, yes. This is the part you should remember. And one more thing. This is what? Nuclear envelope. Okay, this is the nuclear envelope of the cell. And this one a thread like structure is there at the center of the nucleus called chromatin. Okay, chromatin. This one also got duplicated during the S phase because of DNA replication. Okay, already it is doubled now, but not having individual identity as the chromosomes. It is in the form of thin thread like. Now we call it as a chromatin, not chromosome. When it becomes condenses and took the shape of chromosome, then we will call it as a chromosome. Now it is chromatin right now. Okay, and one more part here is there in the cell, that is, okay, can you name this organelle which is located inside the nucleus? Yes, can you name this? Yes, you are correct. It is nucleolus. Nucleolus. Okay, and in the cytoplasm there might be other cell organelles such as endoplasmic reticulum. Okay, here, see here, this. Now I am not telling regarding prophase. Before prophase, what are there in the cell? I am telling. Okay, see, it is a endoplasmic reticulum, and this one what? Mitochondria. This one what? Golgi apparatus. Like that. So many cell organelles are there. Okay. Now this is before the cell enters into the prophase. The cell appears something like this. When cell enters into the first phase, that is prophase, what changes will occur in the cell? I am going to tell. Please observe it. Okay, see here. When prophase begins, this nucleolus gradually disappears. When prophase begins, what happens? The nucleolus gradually disappears, and even the other cell organelles will also disappear. Okay, at the end of the prophase, no cell organelles such as mitochondria, Golgi apparatus, endoplasmic reticulum are visible. Okay, so these changes will occur in the cell. Along with that, the nuclear envelope will also get disintegrate or disappear. Nuclear envelope will also get disintegrate. In the meantime, the main important change regarding prophase is this chromosome will start getting condensing. And it took the shape of chromosome. Okay, so chromatin will gradually turn into what? Chromosome. Okay, now they start getting condensing. Something becomes like this. Okay, chromatin, something become like this. See, I am showing chromatins. Chromatin will become into what? Chromosome. They start getting condensing. Yes, like this. Okay, but they not fully get condensed. Okay, they fully get condensed during metaphase. Now they start getting condensing, but not completely condensed. Okay, that point also you need to remember. These changes will occur during S phase. Along with that, the centrosome in the cytoplasm will also move towards the opposite pole with the formation of a microtubules called asters. Okay, see here, let's assume this is one centrosome with two centrioles. Okay, now it is forming what? Microtubules called asters. Okay, now these centrosomes will start moving towards what? Opposite poles with the formation of microtubules called asters, and along with that a spindle fiber, along with the formation of spindle fiber. This is how both the centrosomes will move towards the opposite pole. Okay, all these changes occur during the prophase. Okay, let us list out the key features of the prophase. Okay, what you noted right now. Okay, let us list them in the form of key features. See here, prophase, this is the first phase of the mitosis or M phase, and second point is chromosomal condensation starts. Okay, chromosomal condensation will begin. Will begin. Chromosomal condensation. This point you need to remember. Okay, during prophase, what happens? Chromosomal condensation will start or begin. Okay, chromatin will gradually turn into chromosome. Chromatin will gradually turn into chromosome. Okay, this important change will occur during the prophase, and the nuclear membrane will gradually disintegrate. Nuclear envelope, we can say nuclear membrane or nuclear envelope. Envelope will what? Nuclear envelope will gradually disappear, gradually disintegrate. Nuclear envelope will gradually disintegrate. Okay, so gradually it disappears. And one more important change that occurs during the prophase is nucleolus disappears. Nucleolus disappears. So during prophase there will be no nucleolus. Okay, here there was a nucleolus that was completely disappeared. And one more important change occurred during the prophase is the centrosome will move towards the opposite poles with the formation of microtubules or asters. Okay, see, centrosome. See here, centrosomes already duplicated in the S phase. Now they start moving towards the opposite pole. Centrosomes now start moving towards the opposite pole with the formation of microtubules called asters. And when they form microtubules, then they are called what? Asters. Okay, so there were two asters right now. Okay, so with the formation of microtubules called asters and spindle fiber, there is an important role of spindle fiber in the cell division. That is, these spindle fiber get attached to the chromosome at a particular point called kinetochore. Okay, then they pull those chromosomes, okay, to the opposite poles. So as to pull the chromosomes, spindle fiber plays very important role. I hope all the key points regarding prophase are clear to you. Okay, if you want to take a screenshot, you can have it right now. Okay, Now let us talk regarding the next phase, that is metaphase. Okay, let's discuss about next stage of the cell cycle, that is metaphase. Right, see this diagram you have to remember regarding the prophase. By looking at the diagram itself you can remember all the key points. Okay, now let us talk about the second phase of the karyokinesis, that is metaphase. Metaphase, by looking at the diagram itself, okay, by looking at the diagram itself we can remember all the key points. Okay, if you look at this diagram you can easily recall all the key points regarding prophase. Now let us make certain changes, okay, in the cell, so that we can remember all the key points regarding metaphase. Okay, yes, in during the metaphase what changes will occur in the cell, let's see one by one. See here the nuclear envelope completely gets disintegrates during the metaphase. See here, during the metaphase, okay, what happened, nuclear envelope completely disintegrates or disappears. Okay, nuclear envelope, there will be no nuclear envelope at the metaphase. This is what one of the characteristic feature of the metaphase. Okay, disintegration of nuclear envelope completely, this is mark the beginning of the metaphase. Okay, how do you come to know that metaphase got started? If nuclear envelope gets completely disappears, then that mark the beginning of the metaphase. So nuclear envelope completely disintegrates. Then what other changes will occur? See here, already centrosomes are moving towards the opposite poles during the prophase. At the end of the prophase or in the beginning of the metaphase, these centrosomes are exactly at the opposite pole. Okay, centrosomes are exactly at the opposite poles. This is also one of the important key feature regarding the metaphase. And one more thing is their spindle fibers got attached. Okay, their spindle fibers got attached to the centrosome of the cell cycle and pulled it and made them to align in a central equatorial plate. Okay, so see here, spindle fiber got attached to the centrosome of the water. Spindle fiber got attached to the centromere of the what, chromosomes. And now in the metaphase all the chromosomes come and lie on the central equatorial plate. Okay, this is what one of the important characteristic feature of the metaphase. See here, let me draw it. It is taking few minutes. Okay, no issues, just wait. Okay, see here, I am using a different color so that you can easily understand the concept. Let's see how the chromosomes came and aligned on the central equatorial plate. Okay, see here, this is considered as central equatorial plate. All the chromosomes aligned in the central equatorial plate, and the spindle fibers from opposite what, opposite centrosome came and attached to the centromere of the chromosome. Okay, let me clear all the points regarding M phase. Okay, see how this happened. Yes, see here, metaphase. This is one of the important phase of the cell cycle. here all the chromosomes aligned at the central equatorial plate, and this is the stage at which chromosomes chromatin will completely condensed and took the shape of chromosome. Okay, now this is the stage at which we can easily study the morphology of the chromosome. Okay, this question might be asked. Okay, what's the question? At which stage of the cell cycle morphology of the chromosome is clearly studied? Then your answer going to be what? Metaphase. Okay, this is the stage at which we can study the morphology of the chromosome. Yes, let us assume this is what chromosome. Okay, here this is a chromosome which is of X shape right now. Even the shape may varies. Shape may varies means like this, like this, or like this. Okay, shape may varies based on the shape. Even chromosomes are named. Named. Okay, that you are going to study in the chapter called cell. Okay, now I am taking one typical chromosome. Okay, here this is what the chromosome which is of X shaped. Here these two are termed as chromatids. Okay, what are these? These two are termed as chromatids. Now they are called as sister chromatids. Remember. Okay, these two are called as sister chromatids because this is the initial chromosome. This is the initial chromosome, blue color I am showing. Let's assume that is the initial chromosome. During S phase, during S phase, DNA got replicated, so that's why its sister chromatid were formed during the S phase. The chromosome content got doubled. See here, chromosome number remains same, but chromosome content get doubles. Okay, this is how it happened. Okay, these are called what? Sister chromatids. And there is a center, at the center of the chromosome there is a structure called centromere. This structure is called what? Centromere. Remember, here there are some words. See here, this is called what? Centrosome. This is centrosome. Okay, centrosome. Centrosome is made up of two cylindrical bodies called centrioles. Centriole. Here the similar word is there. Okay, in chromosome at the exactly center of the chromosome one spherical structure is there called centromere. Okay, this is different from those things. Okay, this is what? Centromere, and these two were called sister chromatids. And one more important thing here is on centromere, on the both lateral sides there were certain disc shaped structures are there. Disc shaped. Those are called as kinetochores. What they are called? Kinetochores. This structure is also very important because the spindle fiber from the spindle fiber from centrosome comes and attaches with these disc shaped structures called kinetochores. So spindle fibers comes and joins to these disc shaped structures called kinetochores. See here we are seeing spindle fiber got attached to the chromosome. Where they got attached to the chromosome? On the disc shaped structures called kinetochores. There they will get attached. Okay, now let's see. This is what the structure. This is what the diagram for representing metaphase. Now let us write the key features one by one. Okay, key features one by one. See here, metaphase. This is the second phase of the cell cycle or karyokinesis, and here one important feature that happened is see, nuclear envelope got completely disappears. Nuclear envelope completely disappears. Completely, okay, disappears. Okay, nuclear envelope completely disintegrates. So this marks the beginning of the metaphase. This marks the beginning This marks the beginning of metaphase. Beginning of metaphase. Okay. Yes. This is one important key feature. Then centrosomes are exactly at opposite poles. Centrosomes are exactly at opposite poles. Exactly at opposite poles. And their spindle fiber are attached to the kinetochore of the chromosome. Okay, already I explained all these points. Okay, and one more important key feature of metaphase is, okay, all the chromosomes come and lie at the central metaphase plate or equatorial plate. All chromosomes lie on central equatorial plate, or metaphase plate. This is the one of the key feature of the metaphase. Okay, very important key feature of the metaphase. Okay, all the chromosomes lie on the central equatorial plate. Okay, this is what one of the important key feature. Okay, how the cell looks on this phase, we can study it with the help of onion root tip experiment. Okay, onion root tip experiment, where we can see all the phases. Even I will show some images regarding these phases. Original image of cell divisions I will show here. Okay, yes, this is how the cell will look like. Okay, right. See, this is what these are what important key feature. One more important feature regarding metaphase is, this is the phase at which we can study the morphology of the chromosome. Okay, on this phase we can study the morphology of the chromosome very clearly. Okay, so I hope all the points regarding metaphase are clear to you. Okay, now let's talk about the third phase, that is anaphase, and what changes will occur in the cell we'll discuss. Okay, see here, this is what the third phase, anaphase. Okay, anaphase. Okay, yes, we can easily write one point, that is, this is the third phase of karyokinesis. And here what changes will occur in the cell we can study. See here, what are the changes will occur? See, the centrosomes pull the chromosomes, the sister chromatid, towards the opposite poles. Okay, due to which what happens, centromere splits. Okay, centromere splits, and the sister chromatid will split, and now they become what, daughter chromatids. And each daughter chromatid will move towards the opposite poles. Each daughter chromatids move towards the opposite pole, so that they will form what, daughter nuclei, future daughter nuclei. Okay, that important change occurs during the anaphase. Let me show the changes here itself. See here, a blue colored sister chromatids, now they are moving towards this pole like this. Yes, see here, the sister chromatid now became what, a daughter chromatid. They are moving towards the opposite pole. Okay, and here this red colored sister chromatid moving towards this opposite pole. This important change occurs during the anaphase. One of the important phase. Okay, this is how the daughter chromatids will move towards the opposite pole. Okay, so what changes happens during the anaphase? Centromere splits and the sister chromatids will get separate, and now they become what? Daughter chromatids, and those daughter chromatids move towards the opposite poles, and they will be in the shape of phi. Remember, they will be in the shape of what? Phi shaped. Right now chromosomes are in the phi shape. Before this they are in the form of X shape. Now they become what? Phi shaped and move towards the opposite pole. Almost at the end of the anaphase they reach the what? Opposite poles. Even one more important change occurs, that is the shape of the cell quite changes. Okay, shape of the cell will also get slightly change like this. Okay, yes, this is what the important changes occur during the anaphase. Let us write the key features one by one. Anaphase is the third phase of the cell division, and here the changes, what the changes occur? Centromere splits, and sister chromatids will get separate. Sister chromatids will get separate and move towards the opposite poles. Now they are called as daughter chromatids. Now they are called as daughter chromatids. Sister chromatids now splits, and okay, they will start moving towards the opposite poles. Now they are called what? Daughter chromatids. So they are going to form a future daughter nuclei. They are going to form future daughter nuclei. Okay, what's the key feature here? Centromere splits. During anaphase, centromere splits and sister chromatids will get separate and move towards the opposite poles. Okay, move towards the opposite poles. Almost at the end of the anaphase they reach the opposite pole. Okay, what is the shape of the chromatid right now? Chromatid are phi shaped. Okay, chromatids are phi shaped. English alphabet phi shaped. This is also an important key feature. So they move away from the chromatids move away from the central equatorial plate. Okay, that is also one of the important key feature of the anaphase. Okay, now let's talk about the last phase of the karyokinases. Okay, yes, let's talk about the last phase of the karyokinases. Okay, that is telophase. Okay, what was that? The fourth phase or final phase, okay, called telophase. This is the final or last phase of the karyokinases. Final phase, during which what changes will occur in the cell? I will show in the diagram itself. Observe the changes in the diagram. See here telophase. During telophase, all the changes that occurred in the prophase will get reversed. Okay, this is what important characteristic feature Okay, you can remember here. See here, all the changes that occurred in the prophase will get reverse. Okay, let us recall what are the changes occurred in the prophase. See here, in prophase, nuclear envelope get disintegrates. Now what happens here? Nuclear envelope reappears. Okay, nuclear envelope reappears. Then what happens? Their nucleolus got disappeared. Now here what happens? The reverse of that, nucleolus will reappear. Okay, nucleolus will what? Reappear. There in prophase, chromosomal condensation occurred. Now that got reverses here. Here chromosomes will decondense. Okay, chromosomes will decondense and become into chromatin. They lose their identity. Okay, so chromosomes will get decondense, again they become thread like. Okay, they lose their identity. Okay, this is what the change occurs in the telophase. Okay, and all the cell organelles like Golgi body, mitochondria, endoplasmic reticulum, they will start reappearing. Okay, all these important changes occur during the telophase. Just remember, telophase is just a reversal phase as that of the prophase. Okay, yes, what are the changes occurs here? Nuclear envelope, nuclear envelope, envelope. Okay, envelope reappears, reappears, nuclear envelope reappears. Okay, so then nucleolus reappears, nucleolus reappears. Then chromosomal decondensation will start. Chromosomal decondensation will start. Okay, and they become into chromatin. They become into chromatin. Yes. So and all other cell organelles will reappear. All other cell organelles will reappear. Yes, this is what regarding telophase. Okay, so at the end of the telophase, two daughter nuclei were formed. At the end of the telophase, two daughter nuclei are formed. Here, then one more important point, what is the ploidy right now? About that we have to concentrate here. In the parent cell there were how many chromosomes? 2n. Okay, here also at the end of the telophase the chromosome number remains same, because okay, the centromere splits during the metaphase. Okay, just recall the points. In the metaphase, centromere splits and two daughter chromatids will move towards the opposite pole during the anaphase, due to which what happens? Chromosome number remains same, but the chromosome content will again come to what? From 4c to 2c. Okay, in prophase the chromosome content was 4c, but during anaphase and at the telophase the chromosome content will become into half, that becomes into 2c, but chromosome number remains the same. Okay, that is why mitosis phase is called as an equational division, because daughter cells are having same number of chromosomes as that of the parent cell. Okay, this will happen during the telophase, and even the shape of the cell also changes. It will take the shape of 8. Okay, the number 8. Okay, the cell will take the shape of 8. Okay, this is what telophase. Now let's talk about the last phase of the cell cycle, that is cytokinesis. Cytokinesis. This is the second phase in the M phase. M phase is divided into karyokinesis and cytokinesis. We studied all the four phases of karyokinesis: prophase, metaphase, anaphase, telophase. Now what we are going to study now is we are going to study cytokinesis. Here cyto refers to cell, kinesis refers to what? Division. Okay, cellular division will occur in the cytokinesis. Okay, yes. Let's see what happens during cytokinesis. Cytoplasm will be equally distributed to the two daughter cells. Cytoplasm and other cell constituents are equally distributed to the two daughter cells. And here in case of animal cell, what happens? A small furrow occurs in the plasma membrane, and that gradually deepens. Okay, small furrow occurs in the plasma membrane, and that gradually deepens from external to inside, and finally joins, and two daughter cells are formed at the end of what? Mitosis. Okay, with the same number of chromosomes as that of the parent cell. This is how the cytoplasm will get divided during the cytokinesis. So here in case of animal cell, what happened? A small furrow occurred in the plasma membrane that deepens from external to inside, and later joins and separates into two daughter cells. Okay, whereas in case of plants, a slightly different phenomena is there. In case of plants, what was that different phenomena? Let us see here. In case of plant cell, there is a rigid cell wall made up of cellulose. Instead of plasma membrane, there is a rigid cell wall. Okay, so here in case of plant cell, what happens? In case of plant cell, no such furrow occurs in the cell wall, which get deepens from external to inside. Instead, at the center of the cell, a small precursor will occur, which is also called as a cell plate. Precursor. It's going to be a precursor, a cell plate, which will later develop from center to outside lateral walls. Central to outside lateral walls, and finally divides the plant cell into two daughter cells. This is how the cytoplasm divides in the plant cell. Okay, this is regarding the cytokinesis. Okay, now what I will show here is I will show all the phases, their images on the screen. You can compare what changes occurred in prophase, then in metaphase, anaphase, telophase, and lastly cytokinesis. Okay, with the help of diagram, you can easily remember all the key features. Now what we will do is we'll talk about the final thing, that is, what is the importance of mitosis? Why mitosis is important? About that we'll discuss. See here there are two main important points you have to remember regarding the importance of the mitosis. That is, one is growth. Okay, see, each and every organism is growing, especially cell growth occurs because of mitosis. Not only cell growth, the organism's growth is occurring because of what? Mitosis. Okay, if we get damaged, okay, if we get accident, or if we get damaged, what happens? Our skin will get rupture. Okay, after few days the skin will reappear. This phenomena is because of what? Mitosis. Okay, then during our childhood we are very small. Right now we are big. Number of cells got increased in our body. That is because of the what? Mitosis. So mitosis leads to what? Leads to growth. Mitosis leads to what? Growth. Okay, and one more thing is repair. Huh? Repair is also taking place because of the mitosis. Any damage or any death of the cells will be replaced by other new cells. That is because of mitosis. Okay, see here, mitosis is occurring in our epidermal cells. Okay, epidermal cells are replaced by mitosis. Even blood cells are constantly being replaced in our body. Okay, RBCs are having, okay, lifespan of 120 days. After that new RBCs are going to form. That is because of what? Mitosis. So blood cells are reforming in our body. Even our gut walls sometimes get damaged. Internal gut walls, okay, their cells are also replaced by mitosis. This is how in animals body mitosis plays an important role. And even in case of plants also, okay, plant growth is happening because of the apical meristem as well as lateral meristem. There also a cell division is taking place. That is nothing but our mitosis. This is what the importance of mitosis. Okay, with this we completed one important one important cell cycle, that is mitosis. One more important cell cycle is there called meiosis, which is going to be very interesting and important as for examination point of view. Okay, if you like this video, please hit a like button, get subscribed to my channel and share it to your all friends. Okay, for such educational videos, get subscribed and hit a notification or bell button. Okay, so that you can instantly get our new updations. Okay, thank you so much.